Welcome to the webinar REVIVO's 4D Organ-on-Chip Technology, Introduction to ReleGO and REVex. In this webinar, we're going to give you an introduction to the features of REVex microfluidic chip and their function. You will also understand what's the fourth dimension in REVIVO's technology and what makes it stand out in the field. We'll continue with an introduction to ReleGO automated fraction collector and show you its various features. The last 15 minutes of this session will be dedicated to any questions you may have, and REVIVO's microfluidic scientist Philip is here with us today to answer them. Please feel free to type your questions at any point during the presentation or ask away during the live Q&A. To type your question, please find the chat icon on the bottom panel, or if you can't see it, click More and select Chat.
Hello, I am Philip, REVIVO's microfluidic scientist, and in this webinar, I'm going to give you an introduction to our 4D in vitro tissue-on-chip platform. It consists of a specially engineered multifunctional microfluidic device paired with an automated fraction collector. Together, this platform can help you carry out all kinds of in vitro tests such as safety and toxicity assays, permeation tests, and efficacy studies for your ingredients and product formulations. Let's begin with our microfluidic device, the REVex. One REVex chip holds 3 miniaturized tissue chambers in which tissue samples are loaded to perform your desired experiment. The chambers can accommodate a variety of skin simulants such as human skin explants, commercially available 3D reconstructed skins, synthetic membranes, and other tissues. Each tissue chamber is independent from one another and fits tissue size of about 8 mm in diameter. We recommend to use samples with a thickness of 1 mm, though the chamber can accommodate thicknesses of up to 2 mm. REVex allows you to work with smaller sized tissues, which can be especially useful when working with rare or hard-to-source real skin explants or other explant tissues. Another advantage is that it allows for the possibility of using explant tissues from the same donor across samples in the same experiment, reducing variability in the results. These are the dosing bars, which affixes tissues in place, preventing the tissues from shifting during the experiment. It also prevents the media in the microfluidic channels from leaking to the top of the tissue. There are two types of dosing bars on REVex to cater to different types of formulations and experiments. The low dosing bar has a capacity of up to 40 microliters and is suitable for finite doses or more viscous formulations such as creams. The high dosing bar takes up to 250 microliters and is suitable for infinite doses or more liquid types of formulations. To apply topical ingredients or compounds on skin or tissues, place them on top of the tissue here. Our platform also allows systemic treatments, which can be applied from the buffer reservoir or through the feeding tubes. The next feature of the REVex chip are the microfluidic channels. They allow the dynamic flow of the media beneath the tissues to mimic the physiological function of the blood and interstitial flow inside our body. This dynamic flow perfuses the tissue with a constant supply of nutrients to keep the tissues viable for an extended period of time. At the same time, it also eliminates cellular waste. Conventionally, static tissue and cell culture environments consist of a static media for which tissues are submerged in. As the media is changed every 2 to 3 days, this results in cellular waste accumulating, affecting the long-term health of the tissues as well as its physiological relevance. In vitro 3D cell culture models can effectively model some aspects of in vivo human tissue physiology. However, they are unable to simulate the realistic and continuous movement of nutrients and waste products within human tissues. Consequently, assessments conducted in such static models may not fully capture the complexities of in vivo responses and could potentially yield results that are less representative of real-world physiological conditions. In contrast, with the presence of microfluidic channels, our dynamic system results in a natural tissue environment that closely resembles that of in vivo-like physiological conditions. The presence of dynamic control is why we refer to our technology as 4D. Therefore, testing of new substances is carried out with enhanced accuracy and effectiveness, surpassing the capabilities of static tissue models that do not replicate the dynamic aspects of blood flow. Another function of the dynamic flow is that it carries away soluble factors that are released by the skin as a result of the topic or systemic treatment. These soluble factors are collected as eluates at your desired time points and can be further analyzed. Besides analysis of soluble factors in the eluates, histological analysis can be performed by removing tissue samples from the chip. Now, let's move on to the automated fraction collector called ReleGO. This is where REVex chips are placed. REVex chip loads 3 samples and up to 4 REVex chips can be used on the ReleGO at the same time. Therefore, ReleGO can run as many as 12 individual samples simultaneously. If you would like to run more than 12 samples at the same time, you may do so by utilizing 2 or more ReleGO units in parallel. The REVex are placed onto the elevated bridge of the ReleGO, which features 2 heating plates responsible for regulating the temperature of the microfluidic systems. The heating plates are calibrated for the temperature of both the tissue samples and the flowing media underneath to reach 37 degrees Celsius, mimicking the physiological temperature of our body. If no heating is needed, this feature can be deactivated by the user during the setup phase of the experiment. The automated fraction collector ReleGO is designed to be intuitive and easy to operate. Via a touchscreen, you can follow the instructions on the menu to choose your desired experimental duration, time intervals, and flow rate. Here, we have a 96 well plates on a moving tray. As mentioned earlier, the soluble factors are carried away by the media to the end of the REVex chips, where droplets are formed and collected at different set time points by ReleGO into these well plates. What is shown here are the standard 96 well plates with a volume of 250 microliters. Where you anticipate higher volume, deep well plates with volumes of 3 to 4 milliliters can be used too. 96 well plates are a staple tool in laboratories and readily compatible with standard microplate readers, the analytical instrument able to detect and quantify biological and chemical analytes inside the solution. Besides analysis of the eluates using microplate reader, outflow samples and tissues collected from ReleGO can also be analyzed and processed in many different ways. From the collected outflow samples, you can perform chemical analysis such as HPLC and gas chromatography, or biochemical analysis such as ELISA and Western blot. You can also gather data from your tissue samples by performing histology, H&E staining to compare morphology of tissues, immunostaining, or omics. By analyzing these tissues after the automated protocol, crucial information about the nature of skin condition and illnesses can be extracted, shedding light on their underlying factors. This analysis also aids in demonstrating the safety and efficacy of your skin treatments. Let's move on to the last 2 components of ReleGO fraction collector. For light-sensitive compounds and formulations, we provide a lid that will block out the light. And lastly, this is the peristaltic pump. This low flow rate pump draws media from a buffer reservoir through feeding tubes and into the microfluidics channels of the REVex chip. Typical flow rate ranges from 4 to 20 microliters per minute. These are the main components of REVIVO BioSystems tissue-on-chip platform, ReleGO and REVex. Let me know your questions during the Q&A sessions of this webinar.